today, I think what we'll do is we'll review the antigen receptor and accessory molecules and focus on the immunoglobulin molecule and how the immunoglobulin is developed or made by the B cells. We'd already talked about, this is sort of a summary slide of the stories that we've been studying. The endogenous antigen is presented to MHC class 1. The exogenous antigen is presented to MHC class 2. Non-protein or non-amino acid based antigens are presented by CD1 to NKT cells. NK cells recognize cells that don't express MHC molecules, and antibody molecules can recognize antigens in its native form. So that's what we're going to look at today. A little bit about Rolf Steinman, who discovered the role of dendritic cells, for which he was given the Nobel Prize. Now the first slide that I showed you was the one that we had spent a lot of time in the last couple of lectures talking about. Now, from henceforth, we'll start looking at this slide as sort of the one slide which talks about various aspects of immunology, where antigen processing takes place in the antigen presenting cell. That antigen that is presented to the T cell. The T cell then activates B cells to make antibodies. And the CD4 positive T cell also helps CD8 cells differentiate into cytotoxic T cells. We'll talk about all this regulation in the upcoming lectures. These are the topics that we are going to go through in this lecture, which is antigen receptor and accessory molecules. This may not make a lot of sense to you right now, or maybe it will, but then we'll review these after the lecture is done. Okay, so now let's dive a little bit deeper into the antibody molecule itself. So I think most of you will know that the antibody molecule is made up of two chains, heavy and the light chain. The heavy chain is the bigger chain and the smaller chain, right? And so you can see on the right side, the brown and the light blue are the heavy chains, two heavy chains which come together, but they're expressed by a gene, a protein that is expressed by a gene, and then the light chain, which are shown here in purple and gray, are expressed by another gene, which is present on another chromosome. And when they come together inside the cell, when they are forming a protein, a collective protein, they form these structures at the junctional region, which are called complementarity determined regions, which, as you can see on the left side, are the antigen binding site of the antibody. The tail of the antibody, so to speak, the Y, the straight line of that Y, are the FC receptor binding proteins. FC stands for, the functional crystalline, that's the official name, but it is the portion of the antibody that binds to receptors on cells that are not necessarily antigen specific. So the antigen specificity comes from the variable domain, which is here on the top, the antigen binding domain. A little bit more about the antibody in terms of structure. These are just redundant slides that showed you different ways of looking at the antibody. Again, the heavy chain and the light chain. You can see here CDR, complementarity determining regions 1, 2, and 3, are the specific sites at the antibody that like if your antibody forms a pocket and the pocket actually binds the antigen, the residue, the amino acid residues that bind to the antigen are called the CDRs. And you can also see that the antibody molecule has a three-dimensional structure. It has all these cysteine residues, which are shown here in black lines, which hold these antibodies together in a structure. Antibodies can either be secreted by the B cell or they can be membrane associated, and this is dependent on the transmembrane region that they might express during the gene expression process. So looking at the structure of the antibody, if you take these styrofoam tubes that are shown here on the right, and you fold them in a particular way, take a long styrofoam tube and you fold them in a particular way, shown here, you can see that the tips of these folds are the portions which are equivalent to the folded structures which form the CDRs, right? So that's CDR 1, 2, and 3. If I had to sort of expand this, take out that rubber band and allow the styrofoam tubes to be flat, this is what you would see. You would see the interspacing between the framework region, which is the conserved region, CDR1, CDR2, and CDR3, which are not continuous on the protein molecule. This protein is encoded by the germline DNA, which has three major components, all variable, diversity, and joining, and we'll look at that a little bit more in detail. And the light chain has the variable and the joining gene. They don't have the diversity domains. So the heavy chain is present on the gene for heavy chain is present on chromosome 14. The gene for the kappa and the lambda light chains are present on chromosome 2 and chromosome 22. This is very different from the MHC molecule that we studied in that previous lecture, where all the genes that are associated with the MHC A, B, C, DR, DP, DQ, the genes for the proteasome, the genes for TAP, the genes for actually DM, etc., everything that is involved in the antigen processing pathway, all the genes are associated in that MHC gene locus, right, which is on chromosome 6. Here, an antibody molecule is made up of multiple different proteins, and each protein is expressed by a completely different chromosome. So the evolution of this protein, of the antibody molecule, it has been very different from the evolutionary process of the MHC molecule. And we may not get it today, but we'll talk maybe in a future lecture of how the evolution of the antibody has come about to our body. And that's an absolutely stunningly fascinating story. We'll come to that later. But let's look at the structure of the antibody molecule first. So from a protein perspective, we saw that the antibody has a variable domain and it has a constant domain. The variable domain of the antibody is encoded by three portions of the gene, which are called V for variable, D for diversity, and J for joining. V, D, J. And the constant domain, which is the Fc receptor, comes from the constant domain called C mu, C delta, C gamma, etc. And you might have heard of these molecules called IgG1, IgM, IgA. You've heard of all of those, right? So IgM comes from a gene that is encoded by the C mu sequence, right? So C mu encodes for a protein, a portion of the protein that we call IgM. C delta is the gene that encodes for IgD. C gamma 3 is the IgG3, etc., etc., etc. And we'll see how all of these subtypes or isoforms of the immunoglobulin molecules are expressed and secreted. So let's look at the VDJ sequences first. There are multiple V's and there are multiple D's and multiple J genes within this chromosome 14 locus. It's a gene duplication process evolutionarily. So in a particular B cell, which is undergoing this recombination of forming the VDJ sequence in the antibody molecule, there is one V which recombines with one D, which recombines with one J. They come together and form this antibody structure. I'll let you see this video offline later on, but let me show you what's happening from a molecular perspective. So I told you there are multiple V genes, multiple D genes, and multiple J genes. In a single particular B cell, which is this B cell is developing the antibody molecule in the bone marrow. So that's where the generation of the B cell is occurring. So now in this example that I'm showing you, a V5, 1, 2, 3, 4, 5, the V5 position is going to recombine with the D2 gene that is present here. Now, it's a random process. So in every B cell, a different V will recombine with a different D. And this, I'm saying V5 and D2 are coming together. To the 3' of every V gene, which is the right side of every V gene, is this sequence called heptamer 23 base pair nonomer. It's called a recombination or recognition signal sequence, RSS. And on the 5' of every D gene is this sequence called heptamer 12 base pair nonomer. And you'll see that there's some similarities to this, because if I show the next slide, you can see it's the exact same slide, but now it's folded. This is the 5' region of the every V gene, and the gene continues to D, and then this is the 3' on the the 3' and the 5' of V5 and D2, and you can see that they are palindromic, CG, AT, GC, AT, GC, etc. So when this palindrome comes together and anneals this molecule, these two separate molecules together, with these differential base pairs in between, it results in activation of a whole bunch of endonucleases and a DNA cutting and DNA repair process, and this is a process where that recognition is the signal for the DNA to be cut. And when that DNA is cut, that portion of the V5D2, that middle portion, is excised, and that portion of the chromosome is gone, and V5 comes adjacent to D2. So that's at the DNA level. And so that's your D5 and G2 that are joining, forming this molecule. Just prior to the addition, so they are cut now, right? Let's say the V5 and D2 are cut, and just prior to joining, there's this phenomenon called N-nucleotide additions, where because there's a DNA repair is going on, nucleotides are added in between those two D5 and J2 sequences to allow them to join more efficiently. Because the N-nucleotide addition is a random process, sometimes you'll get 3 nucleotides, sometimes you might get 5 nucleotides, sometimes you'll get 6 nucleotides, etc., right? So if there is if there's a number of nucleotides that is not divisible by 3, which is a codon, and you get a the RNA polymerase, which will then have to convert this DNA to RNA, will see some missense over there, and that immunoglobulin gene will not make the protein, and if it doesn't make the protein, the cell undergoes apoptosis. So only those cells which survive, only those cells will survive which will have a successful recombination of this event of forming a functional antibody molecule. And of course, during the joining process, there are all kinds of inaccuracies that occur when the joining occurs, and so you'll have all this variability that is occurring. So if you think about what's happening in the B cell, there are millions of pre-B cells that are present in the bone marrow at that time. Each one is undergoing some random recombination. And there's a theoretical possibility that 2 separate B cells will undergo V5 and D2 recombination. But the likelihood that V5D2 recombination will elicit the exact same antibody molecule is very low because of the junctional inaccuracies and nucleotide additions that occur. So it helps to improve the repertoire of the antibody molecule. So there are 45 genes, heavy genes, for 23 diversity genes, 6 joining genes, and multiple different N-nucleotide additions. That gives you a repertoire of 10 to the 11th different types of B cells made from approximately 200 odd gene sequences. So this, I think, is the miracle of the molecular biology of antibody structure, where 10 to the 11 different types of proteins are made from 200 genes. So let me pause here and see if we understand this. I think we made this slide last time. So we said that each B cell undergoes a unique VDJ recombination. So in the example we looked at was V5 and D2. And then there could be a separate junctional sequence. The second point was each recombination can have an increase due to diversity, due to the N-nucleotide addition. So if a V5 and D2 come together, in some genes you might have TGA N-nucleotide addition, another cell will have TGA AGT N-nucleotide addition. So that increases the diversity. Junctional inaccuracies, all of these have to result in a successful productive mRNA for the protein to be made. And this is where we stopped, I think, last time. And let's move on a little bit further. I'm not going to spend a lot of time on the evolution because the evolution itself is a big lecture. But I just want to point out that the first appearance of an immunoglobulin-like gene came in the jawed vertebrates, and the stunningly amazing thing is that the recombination event that took place, which is elicited by an enzyme, which is called the recombination activation gene, RAG. I think if I go back to the previous slide, one of the previous slides, I'll show you here. there's a gene called recombination activation 1 and recombination activation 2. These are two genes that encode for endonucleases, which are responsible for cutting the DNA at that site. RAG gene products are required, right? Fascinatingly, at least for me, the RAG gene was actually a transposon in the earlier non-vertebrates. It was a transposon. Transposon meaning it was an enzyme, nucleic acid regulating enzyme, which cut DNA and allowed the DNA to jump from one chromosome to another. That's what transposons do, right? But that transposon was in this kind of world, the language to use, it was domesticated in the evolutionary process. It was domesticated to be able to use it for the recombination events that take place. With Bhavna Tiwari, who's a professor she was a professor in Institute of Science or NCBS or something, and now she's in Bhubaneswar. She helped me understand this process much better, and we've actually written a manuscript on this topic, on the evolution of the VDJ recombination and how it jumps from being a recombinase to how it jumps from being a transposase to a recombinase. It's a fascinating story. So if you're interested, I can send you that paper. So coming back to where we were of the VDJ recombination, the VDJ recombination events at the genome level result in formation of the VDJ mRNA, which then encode for the VDJ sequence, which is present on the protein, right? So the junctional sequences that are there in between are the ones that cause the formation of the VDJ recombination. If you're interested in the mathematics of how the 200 genes become 10 to the 11, there's a very nice paper in Frontiers of Immunology, which talks about this, a lot of statistical analysis of this. This is beyond the scope of what I can understand at this point, but if you're interested, you can look at this paper. Where do all of these recombination events occur? I had mentioned that they occur in the bone marrow during the development of the immune system, which is early in the stem cell when there is nothing, no immunoglobulin molecule. The first event that takes place is the DJ recombination, diversity and joining recombination event takes place. Once that is successfully completed, then a VDJ recombination takes place, allowing for the formation of the heavy chain. Then you have the light chain recombination, heavy and light, and then you get the formation of a full heavy and light chain, and that forms the antibody. So the question is, what light chain are they expressing? They're expressing the IgM. The first B cell is expressing the IgM because the first constant domain gene, if you see in that, I'll show you that again, is c-mu. And so when c-mu is seen by the RNA polymerase, you get the VDJ and the c-mu, and that's why all immature B cells express IgM, right? Leroy Hood had done a phenomenal amount of work earlier in the 1980s to decipher this whole recombination events that took place for VDJ recombination. I think worthy of a Nobel Prize himself, but I don't think he's got the Nobel Prize for this. But he got the Order of Merit from Obama. So another person who actually got the Nobel Prize was Susumu Tonigawa. He got the Nobel Prize for demonstrating a phenomenon called somatic mutation, somatic rearrangement. And this is a very unusual phenomenon that occurs in mature B cells after the B cells have left the bone marrow and now have come into the periphery, and they are now present in the lymph node. A phenomenon that occurs, which is after the B cells have been stimulated with a primary, secondary, or a tertiary, let's say, vaccination. Like when we were all vaccinated with COVID, and you get one dose of the vaccine. When you get that dose of the vaccine, that vaccine goes into the lymph node, and it stimulates B cells to get activated. When they get that primary activation, that activation signal itself induces an endonuclease, which induces mutations in the CDR regions of that B cell, again in a random process, right? And so that one stimulation induces a few mutations, two stimulations will induce more mutations, and three stimulations will induce even more mutations. And so why is this occurring? This is occurring randomly for the B cells to allow the maturation or an increased affinity. Oh, I remember, we've seen this video last time. I'm not going to see the video. Pushpam, I think you can watch this video on YouTube later on. But essentially what happens is when an immature B cell first gets activated, it induces somatic mutation, and the surviving mutated cells, which have a higher affinity for the antigen, for the limiting antigen that is present, allow for proliferation only of the high affinity mutation. So over a period of time, you get selection of high affinity antibodies upon multiple antigenic stimulation. This process is called somatic mutation. The discovery of this phenomenon was done by Susumu Tonegawa, for which he got the Nobel Prize. Coming back to the structure of the antibody molecule again, this is your heavy chain. We looked at how the heavy chain is formed with the VDJ recombination, the light chain, and the CDRs. Now let's now move in this lecture to the FC portion, to the constant region. So the constant region, this is the lower portion of that antibody molecule, and just to show you the structure, it has this FC receptor binding protein. FC, there are multiple different types of FC receptors, which increases the complexity. This is a phenomenon which is a very important phenomenon in antibody molecules. This is what happens. This is called FcRn. There is a receptor called FcRn. N stands for neonatal. FcR is Fc receptor, neonatal. This is expressed in all endothelial cells, which is the cells of our body which line our blood vessels, right? All of these express FcRn. So what happens is when circulating antibody binds to antigen and binds to the FcRn molecule, it is the antibody molecule, the antigen, everything is taken up through the endosome. You can see all of that FcRn, the Fc receptor, the antibody molecule, and the antigen is all taken up. And in general, the pH in our body fluid is around pH 7. In the endosome, the pH becomes pH 5, and at that pH 5, the antigen separates out of the antibody molecule. The antigen gets degraded and goes into the lysosome. This is an antigen. Remember that antigen processing that we were talking about, when we were looking at antigen presentation, MHC class 2. The antigen is processed and eliminated, but the antibody molecule, because one, it is bound to the FcRn molecule, it is not degraded in the endosome, it actually flips over and allows this antibody to be released without getting degraded. So this is a phenomenon that occurs only in two proteins in our body. One is the antibody molecule, and other is albumin. So these are FcRn binding proteins. And what this does is, because of this property of antibody molecules, the half-life of antibodies is very long. And that is why antibodies have become therapeutic drugs, because of this phenomenon. So if you take any other drug, if you take any other protein, take erythropoietin, growth hormone, or any other protein, right, which doesn't have FcRn binding, it will enter the endoplasmic reticle, it'll enter the lysosome, and it'll get degraded completely, as has been shown here for the antigen. There's nothing to come back out, but the antibody molecule comes back out, allowing it again to bind to new antigens and continue this process of complete degradation, right? So this FcRn property is a very important property of antibody that makes antibodies drugs, antibodies as drugs. And that FcRn binding site on the antibody molecule is shown here in red. That's where that's where antibody molecules bind to FcRn. Yes. Okay, let me pause here because I've been talking quite a bit. Let's do question and answer for a bit.
Sorry for the question.
No. This is good. This is good. I'm glad you stopped me.
Yeah. So this was very interesting. The antibody not getting degraded in the lysosome. So I was thinking, in lysosome generally it's very acidic condition, right? So most of the moieties which are degraded because of the pH. So what makes this antibody really I would say
resistant
different.
resistant
Yeah, exactly.
resistant to
Exactly.
enzymatic cleavage. Pushpam, I thought you were going to ask me that question. I have no idea why they don't degrade, but my assumption would be that, either because of their physical structure and their highly glycosylated nature, they're very, very glycosylated, right? It probably prevents the enzymatic reactions to allow them to be degraded. That's one, could be one reason. The second is, like I showed you in that picture, the antibody is actually bound to the FcRn. So FcRn is the chaperone. It doesn't allow the antibody to be separated from the molecule. So it's not free floating in the end. It's not free
Okay.
floating in the lysosome, and therefore
Okay.
maybe that's why it doesn't get degraded. So this is
Yeah.
all hand waving. There might be actually better reasons. So
Yeah. That makes
yeah,
sense.
but at the end of the day, they don't get degraded. But
Yeah.
ultimately, they do get degraded. It's not like the antibody stays
Yeah.
there forever. It has a half-life of 2 weeks. But why at 2 weeks does it get degraded, and why at 2 hours it doesn't get degraded? I don't have an answer to that.
Yeah. The reason I was thinking, because I was also a chemist. So from that perspective, it looked like, pH is very high, so it should be degrading. But that's very interesting that a different kind of receptor is actually helping the antibody to be more stable in that environment.
Correct. I think if you can do some research and find out, why this might be the case, well, that might be an interesting add to our discussion here. I was going to tell you something. Yeah. So I think I have a few slides later on to show you this. But what people now do, I've been doing it for a while, is because the property of because of this FcRn property of this antibody molecule to increase the half-life is present in that Fc portion, right? What people do is they take just the Fc portion, right, and stick anything onto top of it, completely unrelated. They will remove the antibody portion. You want to take, let's say, growth hormone, and you want to increase the half-life of growth hormone. You stick growth hormone with the Fc receptor, and the growth hormone will have 2-week half-life because it goes through the same um,
Are you talking about
FcRn process.
nanobodies, like people sometimes okay.
Yeah, they can be nanobodies. They can be all kinds of weird, you
Okay.
know,
Okay.
the immunogenicity to these molecules is a whole different story because these are not natural molecules and they look foreign to the immune system. So,
That's true.
so that's another. We'll talk about that separately. But yeah, the take-home message from this talk today is FcRn.
FCN, yeah,
Very
that domain.
important, very important property of an antibody molecule.
I have one more question,
Yeah.
I think, which you might have explained last time. So you mentioned that how in each B cell there are different combinations happening, right? I mean, different B cells have different combinations. So I was thinking, like, in one particular type of B cell, or one B cell, if we take the example, why there is not different combinations happening?
Very good question. It is a process which is termed at allelic exclusion. So
Okay.
once a recombination of one V5 occurs with a D2, right, then there is an enzymatic process, and I don't understand the molecular biology of this, but there is a process which then inhibits any other recombination to take place in that B cell.
Okay.
The term to search for is allelic exclusion.
Okay.
Because
Interesting.
there are two chromosomes, right? Two chromosomes of chromosome 14, and
Yeah.
you can have, in principle, you can have one VDJ recombination occurring on one chromosome and another VDJ recombination happening in another chromosome. So you can have V5 D2 happening here in the other chromosome, let's say D3
Yeah.
D2 happening, something, right? But doesn't it happens only on one chromosome? And once it happens on one chromosome, all other recombination is blocked. And I don't know the mechanism of how that is blocked, molecular mechanism. I know people know about it. If you want to
Okay.
search how that is, the term is allelic exclusion.
Okay, I'll look for it. So
Very
all
good
these
question.
B cell like recognition sites and like not B the antibody production and everything happens when the immune system is growing. So it, like, at very young age, right? Like maybe like few months or something.
Yes. But the most activity of the developing B cell is when at the children's stage, right? Like when 2, 3, 5,
The
10 years
children's.
old, right? At the child. But as the bone marrow, as we grow as adults, our bone marrow, even though it's the extent of activity reduces dramatically, this recombination is happening even now in our body.
Okay.
But at a maybe at a 99% of it is gone, but we have that 1% capability of making new naive B cells. Yeah, Praveen.
Yep. Go ahead, Praveen.
Yes, sir. Actually, as you were talking about FcRn, does this FcRn are present in all the cells of body, like, or only particular cells?
Yeah, good question. So not all the cells of the body, but there are many different kinds of cells that FcRn is expressed in. One of the major cells that is expressed, the reason it's called neonatal, is the placenta. Because it is this expression of FcRn of the FcRn receptor, which is present in the placenta, that allows IgG molecules to go from the mother to the child, or from the child to the mother. Because otherwise, the child will not be protected. The child doesn't have a capability of child's B cell in the womb has no capability of making antibodies. They're not mature enough to make antibodies. So it needs antibodies to protect itself. So that antibody comes directly from the mother, transferred through the placenta. And this FcRn enables that movement, allowing physical movement of the antibody from the mother to go to the child.
Okay.
Yeah, and so that's where the name comes. But it is also present on endothelial cells, which allows it to keep this half-life.
Okay. Other than this, like, because as antibodies are heavy molecules, it cannot enter the cell membrane of cells. So other than FcRn, there is no other antigen presenting or antibody uptake happens.
Yeah, no other antibody. Yeah, antibodies cannot enter the cell. There are not just FcRn. I'll show you this in the next few slides. There are many Fc receptors, but antibodies cannot. They don't enter non-specifically. They enter through these receptor interactions.
Yes, sir. Okay. Thank you, sir.
Good questions. Okay, moving along. Let's see if we can do this. I don't think we'll do it. I'm trying to see if we can finish this antibody lecture today, but we won't. We'll actually need another week to finish it, because different level of complexity, and the topic is complex, right? So it's not a simple. No need to just ram everything just because we have to. We'll come to this. I think there are many intelligent ways that people have been used of making different kinds of antibodies. So, for example, I'm not going to go through this, just to give you a primer, that it is very difficult to make antibodies against HIV, which are neutralizing, which are broadly neutralizing. So people have used this recombination process to understand how to graft CDR3 molecules within a gene to make antibodies like this. So this is a HIV story. And based on this, people are making designer antibodies based on how they can bind. With use of AI ML, people are now able to use that technology to create antibodies in silico. So this is another type, designer antibodies. Okay, moving on to our regular lecture. We started off by saying that B cells are made in the bone marrow. They make resting B cells make IgM. When they come into the periphery, they're exposed to antigen. When they're exposed to antigen, they get activated. That activated, I told you, induces mutations in the CDRs, which causes affinity maturation, which allows them to make high affinity antibodies, right? They also undergo something called isotype class switching, which means from IgM, now they'll start making IgG antibodies. So how does that happen? So that happens by a phenomenon, which is again called recombination. But here, the recombination is slightly different. So just focus on this middle picture that I'm showing you here. You have now got a recombined, you have B cell which has a recombined VDJ sequence. That's in that unique B cell. 3' to the J sequence is this constant domain, long constant domain that you can see, right? You can see C mu and C delta. C mu encodes for the portion of the gene that encodes for IgM. Right, and C gamma 3, for example, here, or C gamma 3, for example, here, will encode for IgG. So what's happening from a DNA level? So when this DNA has to be converted to mRNA, your RNA polymerase will come and bind to this gene and start rolling over this DNA and start encoding for the VDJ sequence. Right? Once it starts encoding for the DNA sequence, it continues to roll, and then it sees the sequence of the C mu. It will start encoding for protein, that is mRNA, that encodes for IgM. At the end of the C mu and C delta gene is a stop codon, right? Well, because the stop codon is there, that's why the cell makes IgM every time. It'll not make IgG3, it'll not make any other gene, it'll stop here, okay? Now what happens is, when the B cell in the periphery has received that initial stimulus, that stimulus of the B cell will activate a whole series of transcription factors. These transcription factors, which the B cell secretes or induces based upon antigenic stimulation, will bind to the S, which is the signal sequence of gamma 3. So remember, B cell being stimulated with an antigen, that antigen will activate a whole series of signal transduction, which will activate a transcription factor. That transcription factor will go and bind to S gamma 3. When S gamma 3 transcription factor interaction occurs, just like the VDJ recombination, there is a fold that occurs, there's a folding that occurs that deletes this portion of the mu and delta chain. You can see the middle, that same portion is deleted, and now you have VDJ and gamma 3. Now this B cell starts making IgG3. It used to make IgM. Upon antigenic stimulation, it starts making IgG3. So when does it make IgG3 versus IgG1 or anything else? It depends on the nature of the signal. If the signal is strong and you have a particular set of cytokines, like say for example interferon gamma, that is secreted to the T cells, interferon gamma and the antigenic stimulation will result in transcription factors which bind to S gamma 3. If the B cell is stimulated in such a way that the antigenic stimulation is of a different affinity, and the T cell secretes, let's say, IL-4, some other cytokine, not interferon gamma, but the T cell secretes IL-4 in the environment, that will induce a different set of transcription factors which might bind to the C alpha, the signal domain of C alpha. And if the transcription binds to C alpha, then this whole portion in between, from C mu to C delta to C gamma 3, and all of them are deleted, and now the B cell starts secreting IgA, if alpha is IgA. Right? So the nature of the signal determines the isotype that you will secrete. Does that make sense?
So in this, I have a question. In this case, why isn't the stop codon working? Is it done stepwise, or is it in the picture it's showing that okay, one step?
Yeah. Let's see if I can understand your question first. First of all, let's start with the first things first. VDJ recombination that you know, right? That we just talked about. And this is the germline sequence. The first sequence that you have is C mu. That's why you make IgM. Okay. At the end of IgM is a stop codon, and that's why this B cell will make only IgM. Now this B cell, which is making IgM, is going to be stimulated in the lymph node with an antigen which binds IgM, and that antigen will induce stimulation of a transcription factor, and that transcription factor will bind to IgG3, to the signal sequence of IgG3. It's like the promoter of IgG3, right? When that transcription binds to the IgG3 signal, it elicits a recombination event to take place in the immunoglobulin gene, whereby this C mu C delta portion of this DNA coils, like it's shown here, just like the one that we saw for VDJ recombination. It is cut by the endonuclease, and that portion is deleted. And therefore, now this B cell, the RNA polymerase, is now reading the immunoglobulin gene, VDJ sequence. Now it sees C gamma 3. At the end of C gamma 3 is a stop codon. Right? That's why this B cell will now secrete IgG3.
Okay.
And C alpha, you can see, is at the end of the chain, right? It is like the last C alpha. You have C gamma 3, gamma 1, 2B, 2A. epsilon, and C alpha. Alpha is encodes for IgA, right? So in order for B cells to make IgA, this entire sequence in the middle has to be eliminated. So this C alpha has got to come adjacent to VDJ. So what happens? Some B cell activation signal would induce a transcription factor which binds to the S signal sequence of IgA of S alpha, right? When it binds there, when it binds to S alpha, this entire portion in between will undergo this fold. It'll get deleted through by the endonuclease. Now your VDJ is adjacent to the C alpha. You see that? You see how that is adjacent to it? And C at the end of C alpha is a stop codon. So now when the RNA polymerase comes and reads the VDJ sequence, it sees C alpha. So you will have a antibody molecule which has a C alpha FC receptor. And that's, IgM, IgG, IgA, and IgE have different structures of FC receptors. It gets more complex of what the functions of these. Why do we have different kinds of subtypes of IgG 1, 2, 3, 4, and IgA and IgE? Because each one of them has a slightly different function. Some neutralize viruses better, some opsonize, which means they coat the antigen a little bit better. Some can activate NK cells, some can activate mast cells. So all the FC and FC interactions are mediated through this antibody molecule, and they bind to different kinds of FC receptors. There's FC gamma 1, 3A, 3B. The story becomes very complex, right? You have IgG 1, 2 antibody molecules, and you have a whole bunch of FC receptor molecules. Those interactions determine the nature of the signals that the B cells receive. And we'll do one more thing, and then we'll close for today, is to see how has the field started using antibodies as therapeutic drugs. So in the 1980s or 90s, when the first monoclonal antibodies were developed for making drugs, you had murine monoclonal antibodies, so mouse monoclonal antibodies. So you would take an antigen, you would immunize a mouse, the mouse would make an antibody, you use that antibody to make the drug. And these are the names of drugs. I'm not even going to try to pronounce them, but these are the names of the drugs that were used as antibodies. They were mouse antibodies to be able to treat particular kind of diseases. And the suffix, the naming nomenclature for these antibodies is the last 4 letters, OMAB. OMAB means they are fully murine antibodies. Then what happened is, and the immunogenicity of this molecule is very high, as you can imagine, because you have mouse antibody given to humans, right? Now, to be able to reduce the immunogenicity of the molecule, what they did was they took just the antigen binding portion of the mouse antibody, and they grafted it onto a human backbone of the FC receptor, right? Now only the top yellow portion is the immunogenic portion. So you reduce the immunogenicity potential of these molecules. And some of these popular antibodies that you know of, rituximab, infliximab, cetuximab, all these drugs make billions of dollars right now in the market. These are called XIMAB, rituximab. These are molecules which have their, the FC, the VDJ recombination portion, the entire domain is taken and put there. Zumab is sort of similar to XIMAB. The difference is, instead of taking the entire domain, you only take the CDR sequences. Only those sequences are taken, and that's called ZMAB. And UMAB are fully human antibodies. They have no mouse component, right? So that's sort of the definition of these. And how do you make fully human antibodies? And there's this technology called xenomouse technology, and different companies have different kinds of these mice. And what has happened is the mouse immunoglobulin gene locus has been inactivated and replaced with a human immunoglobulin gene locus. So when you immunize these mice make human antibodies. Quite ingenious, right? There are other ways to make this antibody through molecular cloning. I was telling you earlier that the FC portion of the antibody molecule is a very important structure because it binds to FcRn. So people take this FC and bind it on all different structures and functions, and you can make all kinds of things. All right, I think these are all just questions that people have asked, and we'll stop here. Great. We'll stop at this last person. We started off by talking about Ralph Steinman, and we're going to end by talking about Max Cooper. The Max Cooper, you can see his paper published in 1965. Now, B cells, functions of the thymus and the chicken. So you know why the B cells are called B cells? Because they were first discovered in the bursa of fabricus in the chicken. Bursa of fabricus is a tonsil-like immune structure in chickens. We don't have it in humans. It's called the bursa. And Max Cooper discovered B cells with Robert Good in this bursa, and that's why they called it B cells. And Max Cooper, is in University of Alabama today. He studied in Mississippi, Tulane, member of the Academy, both in the US and in London. In 2019, as recently as 5 years ago now, he won the Lasker Award. Lasker Award is the equivalent of the Nobel Prize in the US, right? So he won the Lasker Award in 2019 for what? For discovery of B cells, which he had done in 1965. So people get recognized much later in their career. So that's sort of a high-paced, fast story about the B cells, immunoglobulin gene descriptions, and Fc receptor, and how antibodies are used as drugs. So that's today's story. I think hopefully you understood some of it.
Very interesting. I don't know whether it makes sense or not. So, like you said about the immune system, especially in kids and children. So, is that the reason why kids and children they get vaccinated very early in the
good. Yeah, they need early vaccinations because they are not protected. They can't be protected. Okay. They're quite immune suppressed. I have a whole presentation. If you're interested, we can talk about that. Immune responses in children 0 to 2 years of age. Or even in the womb, how the immune system evolves, what is the nature of the immune response in children? Very different.
Okay, I see.
Yeah, Praveen.
So, whenever the antibodies are produced, like it is completely produced with variable region or concentration, or just only variable region is made, because I have read a couple of papers, and they used to mention FC regions are only the variable regions are they are produced, and constant regions are they are constant, they are same throughout all the human beings.
Yeah, so variable domains are different. There are 10 to the 11 different types of variable domains in each one of us. Yes. Then from those 200 genes, but the FC portion is exactly the same in all of us.
And the concentration?
Yeah, same. The all, in that slide where I showed you the green and yellow for the murine and green for the human portion is the same. We have the same, all of us have the same gene. The only difference is the variable domain.
And one general question, why do people get the enlargement of lymph node, whether it is because of the vaccination or because of the antigen which are being invaded by?
Right. So good question. So we'll look at that when we look at immune regulation. But essentially what happens, the lymph nodes that are present are your reservoirs of all your T cells, B cells, dendritic cells, all the cells of your immune system are sitting here. They're like the factory, right? Mm-hmm. And when you get infected or you get vaccinated, then the antigen enters the lymph node. When the antigen enters the lymph node, they start recognizing the immune cells start recognizing these antigens, and they'll start proliferating. And they proliferate hugely, right? Then from one cell, you'll get 1 million cells. And when there's proliferation of these cells, that's why you get your lymph node glands increasing. So because of proliferation, but ultimately when the antigen goes away, they'll all come back to baseline.
Mm-hmm. So how long it takes, just to curious?
Yeah, two weeks to a month. usually it takes for the immune system to sort of resolve any infection. If in a normal situation.
Okay.
But it's a war, right? The viruses and bacteria and all, they don't run away easily. They try to persist for a long period of time.
Yes,
So,
sir.
yeah, but the main immune response occurs within a month.
Yes.
All right. So great, good questions today. I think we went at a little faster pace than usual, but since this was the second time we were doing it, I think it's, at least for Bhavya and Praveen, a little revision.
Yes, sir. So one more question I had.
Yeah.
So why? What is like a difference? Like why people don't prefer plasma therapy? Because as we have the same antibodies and all, we don't get that much immune response to our body. What is the disadvantage of?
I mean, plasma therapy, we all carry all kinds of crap, right? All kind of viruses, bacteria. We have a lot of stuff that's going on. We can't diagnose everything that we all carry. So, plasma therapy means I'm giving my crap to you. And I think we are giving purified antibodies, but with that, all kinds of other things are coming with you. Again, that's how AIDS was transmitted initially. All people who were getting AIDS in the 1980s, not all, some of them were getting because of blood transfusion. So yeah, it is preferred that you can, if you can make a recombinant protein, if you can make these, like hemophilia for factor VIII, for example, if you can provide these from a recombinant way, a purified way, that is much safer than just giving plasma therapies.
Okay.
So I just wanted to revise the points why we have different, or the amount of immunity we have. So higher immunity, lower immunity, all a game of probability. But so like to start with your single B cell, so it's that probability, inherent probability of how each, like it's made in the bone marrow. So it's that, and then it's the lymph node. So then there's again a give probability, and then external factors, which are how many times, or what's the exposure to infection or vaccines, or that thing.
Yeah. So when we once we finish this immunology basic 4 or 5 lectures of the immunology course that we've from regulation, we can maybe talk about mathematical modeling of what that modeling of the immune system might look like. All of these are mathematical equations of how much the cell grows. You can probably model all of these things in a way that is predictable, with obviously with some errors and variability.
Right.
Yeah. All right, good. I know it's getting late for you guys, but I love the enthusiasm that all of 3 of you join, and others comment saying that they can't join. But we do it, and I like this pace because if repeating it a couple of times also helps. Yeah, there's no urgency of finishing off everything immediately. So next time we'll start talking about T cells.
Okay.
Today was B cells, and then maybe NK cells after that. But T cells next time.
Okay.
All right. Thank you. See you next Friday.
Right.
Thank you.